Thanks very much, Amir. Can you see the slides all right and hear me okay?
Yes, yeah, perfect.
Fantastic. Well, I really appreciate the opportunity to speak with you all today. And the previous speakers really gave also a wonderful setup for this talk. I also am going to talk about chlorophyll fluorescence, but I'm going to sort of pivot from the focus on applications in a laboratory context to talking about applications in an Earth system context and to understanding photosynthesis at large spatial scales. So the sort of basis for this is that chlorophyll fluorescence is the only signal we can measure from space that has a unique relationship to the photosynthetic process. Essentially, chlorophyll pigments are capturing solar photons that energy input is driving an electronic transition into an excited state. And the excited state can then relax through different pathways. One of them is that the absorbed energy can be used to drive photochemistry. It can also be dissipated as heat, or it can be re-emitted as fluorescence. And this fluorescence emission is at a slightly longer wavelength than the absorbed light, and it in a sort of Earth system applications, folks will often refer to this as a solar-induced fluorescence, or SIF. So you'll hear me using that phrase in the talk today. In order to use measurements of solar-induced fluorescence to accurately quantify photosynthesis at the global scale, essentially what we need to do is to bring together the top-down and the bottom-up modeling frameworks that have been developed to study large-scale photosynthesis. This is a figure from a really nice review that brought together some of these different state-of-the-art approaches and demonstrated that they, despite the enormous amount of effort and the amazing sophistication of these frameworks, they still have a very large range in terms of their estimate of what global photosynthesis actually is at any given time. And figuring out how to quantify photosynthesis accurately at this scale is a really important, outstanding challenge. What I want to do today is introduce you to a new model that is an important part of the solution. This is a model that was published just a few months ago, and it is the result of a collaboration between myself and Joe Barry at the Carnegie Institution. So these are the sort of the key features of this framework. This is a model that describes the relationship between light capture, carbon dioxide fixation, and the emission of chlorophyll fluorescence by describing how the overall photosynthetic process works as an integrated system. The structure of this model describes photochemical and biochemical processes that are very widely conserved, and the parameters in the model are designed to accommodate the diversity of functional traits within higher plants. This is a framework that can be used both in a forward mode, where you would predict the physiologic performance based on defined functional traits under particular environmental conditions, but it can also work in an inverse mode, where measurements of physiologic performance under particular environmental conditions are treated as model inputs, and the photosynthetic traits are inferred as outputs. Really emphasizing photosynthetic functional traits because they can be looked at as being reporters that are integrating the way that plants perceive and react to both environmental resources and environmental stressors, both above and below ground. So I'm going to sort of pivot now to talking about how this framework works. The core insight that this model is organized around is the insight that an enzyme called the cytochrome B6F complex organizes the dynamics of photosynthetic electron transport in a way that's conceptually equivalent to the way that the enzyme Rubisco organizes the dynamics of carbon metabolism. These two enzymes have a really important role in photosynthesis because they are rate limiting. And so they act as the kinetic bottlenecks within the electron transport system and within carbon metabolism. And they are the targets of many regulatory processes. So in the model, we've represented both the electron transport system and carbon metabolism in terms of the components of the photosynthetic system that can be rate limiting or rate controlling during steady state photosynthesis. So in this framework, the electron transport processes interact with carbon metabolism through the energy supply and demand. And what is really cool about this framework is that the quantification of these limiting processes provides a new way to quantify the regulatory interactions that coordinate electron transport with carbon metabolism. So the two I'm highlighting here are a process called non-photochemical quenching, acting to balance the excitation of the photoax, and a process called photosynthetic control, which is acting to limit electron flow through the intersystem chain at the cytochrome B6F complex. So these are the sort of core ideas in this conceptual model. And what I want to turn to next is an experiment to sort of give you the intuition about how you can see these interactions in common experimental measurements. This is an experimental data set where I used Pulse-amplitude modulated or PAM fluorescence to examine the light response of photosynthesis in poplar leaves. The experiment was very simple. It involved exposing single leaves to sine waves of light, which are shown here in panel A. And each sine wave lasted for an 8-hour period. And every 10 minutes or so over that period, the leaf was analyzed with the PAM technique. So a saturating pulse of light was applied, as in panels B and C. And essentially what happens when you do this to a leaf is illustrated in this lower row of results. The plots here show the partitioning of the absorbed light in these pulses between 3 competing dissipation pathways. The first is dissipation through photochemistry or electron transport. The second is dissipation through regulated non-photochemical quenching to heat. And the third is passive heat loss and fluorescence. And what this shows essentially is that when the excess light is applied in the flash, there's much more light than the plant can actually use. So only a very small fraction of that light is used to drive electron transport within the flash. Most of the light from the flash is being dissipated either as heat or fluorescence, as you see in these panels E and F. This sort of perspective in these lower figures is a little bit unconventional. And the way people would normally visualize these data is illustrated here in the top row of results. So these are back looking on our 8-hour experimental time courses on the x-axis, and looking now at the fluorescence measurements referenced back to the steady state fluorescence. So in this perspective, you can see the part of absorbed light that's driving steady state electron transport, the part dissipated through regulated non-photochemical quenching. And in this steady state, these two regulated processes are compensating with one another, such that only a little bit of the total light is coming back through passive heat dissipation and fluorescence. These patterns are characteristic of how higher plants respond to light. And you can see, even with your eye, the relationship between the fluorescence-based estimate of electron transport in the top left and a gas exchange-based perspective on the same system. So this lower row of panels shows gas exchange measurements that are paired to the chlorophyll fluorescence, and you can immediately see the correspondence. The way that these kinds of measurements are often interpreted, if you're looking just at the PAM fluorescence, people will often look at these and infer that photosystem 2 is doing something or another under different conditions. And people are looking at, the CO2 exchange measurements by themselves. They'll often interpret them as reflecting control by the enzyme rubisco under some conditions or another. And what I want to do today is to give you a sort of a different way of looking at how these discrete parts of the photosynthetic system are interacting with one another. It's sort of a different perspective on the control of photosynthesis. So in this figure, I've projected those same chlorophyll fluorescence measurements in a new set of dimensions. So on the y-axis here, we have an index of the speed of electron transport derived from the Gentil equation. And on the x-axis, we have an estimate of the redox state of the plastoquinone pool, which is derived from the QL parameter. Because these measurements are based on the variable fluorescence, which is coming from the antenna of photosystem 2, they're almost always interpreted in terms of the speed of electron transport through photosystem 2 and into the plastoquinone pool, which is the first downstream acceptor past photosystem 2. This is very logical. What's kind of cool, though, is that in the steady state, the plastoquinone pool couples the acceptor side of photosystem 2 to the donor side of the cytochrome B6F complex. And what that means is that we can interpret these patterns in relationship to what is happening at the B6F complex. So this first phase of the light response, where there's a linear increase in the speed of electron transport as plastoquinone becomes more reduced, reflects the B6F complex exerting kinetic control over the speed of electron flow through the whole chain. What you could do is to follow this relationship out to the point where the plastoquinone pool would be totally reduced. And this provides an estimate of the maximum activity, or the Vmax, of the B6F complex in vivo. Now, as you go up the light response, in the steady state, we never see electron transport going this fast. Instead, what happens is that at the light saturation point, feedback control sets in from carbon metabolism and restricts the speed of electron transport to a rate that is one that the activity of the downstream sinks can keep up with. So these two parts of the response, the low light part, where we see the kinetic properties of the B6F complex, and then the high light part, where we see the regulation of B6F are the core parts of the modeling framework we've assembled. So in the last couple minutes here, I want to switch and show you some applications of this framework. First in a forward direction where we're predicting physiologic performance, and then in an inverse direction where we're inferring functional traits. In a forward direction, these are simulations that show the light response of electron transport on the left and gas exchange on the right for simulations with different photosynthetic capacities. What we can also do is extend these simulations to predict observables that are accessible with reflected or emitted light, so reflectance and fluorescence measurements. And the panel now on the left shows the same light response, but how it's expressed in terms of regulated non-photochemical quenching, and on the right in terms of the steady state fluorescence yield. These capabilities are really interesting and exciting in an earth science context because they're accessible at large scales through inversions. And so what I want to do is switch to looking at examples of model inversions. This is one at the single leaf scale where the gas exchange and chlorophyll fluorescence measurements have been assimilated to infer the leaf functional traits. So you can see there's a good agreement between measured and modeled values for both of these parameters. And this table highlights the range of leaf functional traits that we've inferred from fitting the model to these experimental measurements. I don't want to go into the details of this, but I want to point out that what is unique about this approach is that this model only has mechanistic parameters, and yet it is still simple enough to invert. And this is a really important combination because it means that the parameter estimates can all be evaluated independently, for example, with pigment and protein measurements. This is a kind of a cool example to think about scaling with, because we can conceptualize these core chloroplast processes as being collected in containers that are different sizes. And a leaf is an example of a small container, but a whole ecosystem or a whole forest canopy is something you could look at as just being a larger container for chloroplasts. Tower-based measurements of solar-induced fluorescence and of reflected light can be used to characterize the effective fluorescence yield of whole canopies and the way it responds to environmental variation. This is kind of like what we can do in a cuvette experiment with single leaves, but with a much larger footprint. At the moment, it's possible to replicate this to some extent with satellite measurements. The current fluorescence capable satellites are in low Earth orbits and they have relatively sparse coverage, but this is expected to improve dramatically with upcoming geostationary sensors. And one of the exciting and I think important applications of these observations is to reveal large-scale plant responses to environmental resources as well as stressors. So the points I want to leave you with are here. Essentially, to understand global photosynthesis, we really need to bring together the remote sensing-based and the land surface modeling-based frameworks that have been used to date. The pitch for the new model I've told you about is essentially that because it provides new insight into how photosynthesis works, it allows us to make a richer and a more accurate connection to a wide range of observables that are accessible at large scales. And by decoding the information about plant function that is in chlorophyll fluorescence, we have an opportunity to better understand large scale plant responses to environmental resources and stressors. And I'm really thinking in particular about things like elevated carbon dioxide, about temperature, nutrients, water, the whole range of environmental drivers here. So the model is described in this article, and the code is on GitHub. And if we have any time left for questions, I'd be delighted to take some.
Thank you very much, Jennifer. That was very interesting talk and very nice model that you described. Let me see if we have let's just have one question, and appreciate if you just answer briefly, then people have time for a very short break. So the question is that, very exciting talk. I'm interested in the idea that variation in functional traits from higher plants can be accommodated, detected within this modeling context. I'm wondering about the tremendous CO2 uptake by green algae and cyanobacteria in aquatic systems. With the slightly different photophysiology of these organisms, would the model need to change?
That's a fantastic question. Because I'm a terrestrial person, I've been talking about global photosynthesis, and I meant global terrestrial photosynthesis. But chlorophyll fluorescence can be measured over the oceans. It has been used to study the patterns and controls on marine productivity, but the modeling frameworks that are used to interpret it are quite different than the ones we have for the terrestrial component. And the question that you've posed is right on the nose, because the problem of interpreting the controls on marine photosynthesis, I think, is a lot harder than the terrestrial problem. The functional diversity of photosynthesis in the ocean is much greater than it is on land, and we would absolutely need to adjust the structure of the model to accommodate that. Particular example is accommodating aquatic CO2 concentrating mechanisms. On land, we have things like the differentiation between C3 and C4 photosynthesis. And to accommodate that on land, we do need to change the structure of the model. But with those kind of structural accommodations, we should be able to use this basic approach.
Okay, thank you so much. Actually, we got, I think, 2 more questions, but unfortunately, we don't have time for that at this point. Again, we really encourage everyone to continue discussion and ask questions in the Discord platform. So there's a break right now. We plan to be back at 10:10, but I think with the delay that we have, I suggest that we back at 10:12. So we see you all in about 5 minutes. Thank you very much.